Welcome back to the pharmaphorum podcast. In this episode, I spoke with Dr. Isaac Bentwich, CEO and founder of Quris.AI, who discusses how artificial intelligence could, yes, revolutionise preclinical research and accelerate the development of potentially life-saving drugs safely. From organs on chips to the notion of 3D versus 2D biological models, the conversation touches upon the essential limits of animal testing, though many mice have indeed been cured, and why the rare disease space in particular could benefit from these technological developments in research. Tune in wherever, whenever, and however you prefer to enjoy your podcasts, and do let us know your thoughts. As ever, thank you for listening. Welcome to the pharmaphorum podcast. This is web editor Nicole Raleigh, and today I have with me Dr. Isaac Bentwich, CEO and founder of Quris.AI, a company using AI to de-risk and accelerate drug development to deliver safer, more affordable, and more personalized drugs to patients. Welcome, Isaac.
Hi, glad to be here. Thank you for having me.
So today, Isaac and I will be discussing how AI could revolutionize preclinical research and accelerate the development of potentially life-saving drugs. With about 90% of new drugs failing in clinical trials, pharma companies face tremendous financial losses, around $30 billion plus annually. And more importantly, development and delivery of potentially life-saving treatments are woefully delayed. Infusing AI, therefore, into drug discovery is one of the factors that has led to a 30.6% growth rate of the generative AI or Gen AI drug discovery market in 2024. Traditional preclinical safety data generated by methods such as animal testing very poorly predicts clinical safety and efficacy, but AI has the potential to accurately predict which drug candidates will work safely in humans by eliminating unexpected human responses in the clinical phase due to the discrepancy between animal models and human physiology. This expands the realm of possible treatments benefiting patients with a plethora of conditions, including rare diseases, those who are often marginalized due to financial and trial limitations. So, Isaac, to begin with, perhaps you could tell listeners a little bit more about yourself and your journey to today.
Thank you. So I'm a medical doctor by training, and I've been working in this intersection of machine learning or computation with life science over the last 3 decades. I'm the founder of Quris.AI. This is my fourth company, actually, and so I've previously, in 3 previous companies, have worked on different aspects of using computation to make medicine and more effective. Over the years, machine learning and now AI have been an important tenet of the work that my colleagues and I have done in a previous journey, a previous company. My colleagues and I used AI to analyze the human genome, finding hundreds of novel genes and developing cancer diagnostics based on them. So this, as you've mentioned, that Quris.AI, a company that combines AI together with advanced biology, which we'll be discussing. We are providing a better, more personalized, safer drugs using these technologies.
Thank you, Isaac. So, AI isn't a sort of panacea, as it were, and indeed it has to be focused, which is very important also within clinical trials themselves. So why does most AI-driven drug discovery only show success in one area?
Well, that's a good point, and actually it ties back to your introduction. Traditionally, AI has been used in pharma more on the discovery side than on drug safety, actually. Different aspects of drug discovery have been for the last 10 years or so with growing rates of success been harnessing artificial intelligence or machine learning to do a better job at discovering new targets, new compounds or new drug leads, new target compound fit, reuse of existing compounds. These are all aspects of coming to a drug candidate, which then needs to go through the other processes of drug testing and so forth, including safety. We came into this journey with a focus on an elephant in the room, in a sense, a big unsolved problem of drug safety, which is only a part of the problem, but a really important one, and one which until now has not been adequately addressed. And so that's been our focus.
So safety behind discovery, as it were, which seems bizarre, as you say, very much an elephant in the room. So if we turn to our thematic question, how can AI be used to de-risk and accelerate drug development to deliver safer and also more affordable drugs tailored to
Right.
patients' individual needs?
Right. So again, let me take a few steps back here to paint the picture clearly for us. We spoke about drug discovery, and then we spoke about safety. So the numbers here are striking. The first few years of developing drugs, which is drug discovery, use various methodologies, and AI has come to the rescue and has done a really powerful job using various methodologies of different companies of shortening the time and reducing the cost of finding viable, promising drug candidates. But after you've discovered a new drug candidate, as I've alluded to earlier, then come the next steps. First, testing it what is called in vivo, and then in vitro, testing it in the lab, and then testing it on animals, and then moving into clinical trials. Now, the striking statistics here is that after the discovery process has ended, after several years and several millions of dollars of discovering drug candidates and finding and testing them in animals, and then believing that they're good candidates, you go into clinical trials, into actually testing it into clinical trials. Imagine, though, realizing that the notion that out of all of the drugs that go into clinical trials, after several years and several millions of dollars, and all of that has transpired in the first part of the journey, out of all of the drugs that go into clinical trial, 92% will fail in human clinical trials. This is a horrendous statistics. It is akin to, think of a different industry. Think of real estate. You want to build a skyscraper, you go to the best architect, the best engineer in the land, and you say, I want to build a skyscraper. And the architect tells you, you've come to the right place, here's what we'll do. I'll design 10 buildings for you. I will contractually guarantee that 9 will crumble. We do not know which ones. Just build all 10 buildings, let 9 crumble, and increase the price of the last remaining building. As facetious as this may sound, this is actually the current state of drug development. There is no good predictor right now for which drugs will actually be safe and efficacious in the human body, and even less so for a specific individual. 2/3 of the failure in clinical trials are because of efficacy, a third are because of safety. We've chosen to focus on the lower hanging fruit, the simpler problem, which is still very significant, of doing a better job in assessing which drugs will actually be safe in human. Until now, the approach has been primarily relying on animal testing. That was the best we could do, right? You test it in the lab, and then you want to know whether the drug will be, let's talk about safety, will be safe for use in humans. Why test it in animals? The hope was that it gives some sort of prediction of how it behaves in a mammalian body, how it is metabolized by the liver and excreted by kidneys and absorbed by the gut, etc., how it behaves in a mammalian body. Problem is that we're very different from animals, from mice and other animals, and hence even drugs that pass the animal testing, still the majority, 92%, fail in the human body. And so we are harnessing AI together with advanced biology, as I'm sure we'll talk about, to do a better job than what was currently available in order to predict a drug safety better than was possible until now. And as you alluded with your question, this results both in shortening the time of drug development, drastically cutting the cost, and also being able to address, to accelerate and enable drug development for rare diseases. Why rare diseases? Rare diseases until now have been underserved because the drug development process is very ineffective. So you can justify doing this process, which 90% fails in clinical trials, if you're doing a blockbuster. But if you're doing so for a rare disease that has a much smaller market share, it's not even possible in many cases to develop a drug because the market does not justify the cost associated with developing the drug, because drug development is tenfold ineffective. 90% of drugs will fail in clinical trials. If we're able to reduce that number to make drug development more effective, that allows addressing all of a sudden rare diseases and adjusting them to specific patients.
Thank you. So, yes, as they say, a lot of mice have been cured, but not enough human. So,
Exactly.
if we focus on this notion, and perhaps it wasn't the best applicability of the term lower hanging fruit of safety when in comparison to efficacy, yes, but safety paramount, obviously, when it comes to human trials. If we focus on that safety, I'd just like to sort of tunnel down into the specific ways perhaps that AI can help avoid perhaps drug toxicity troubles.
Right. So drug toxicity and drug interaction and drug personalization. Remember that when speaking about the drug safety, we're interested obviously not just in whether a drug is safe for a human, which is a different thing altogether than safe for a mouse. As you've mentioned, we've cured way too more mice relative to humans, but also safe for who? A drug which is safe for me may not be safe for somebody else because we are all different. And so when we approach this, we looked at two novel approaches that started becoming available and came to the realization that combining them will be an even better result. And these two approaches are AI on the one hand and a form of advanced biology called organ on chip. There is a discipline within biology over the last several years of creating miniaturized human organs in a microfluidic chip, a tiny brain, a tiny beating heart. Each of these organs is typically less than a millimeter in size or something of that order of magnitude. So really tiny version of a human organ, of my heart, of your brain, etc., placed in a microfluidic chip. These were two approaches that are available for drug safety, and both have been showing some promise. These organs on chip, or they're also called organoids or microphysiological systems, these are all have slightly different meanings, but synonyms of these tiny three-dimensional miniaturized human organs on a microfluidic chip, have been shown to be much more predictive with regards to safety than the old two-dimensional biology. By two-dimensional, I mean the classical biology when testing for safety on liver, for example. The methodology was to take the main cell, hepatocyte in the case of liver, and grow it flat, like on a petri dish, hence two-dimensional biology. It does not convey the complexity of the organ because for example, in three-dimensional organs, we have multiple cell types woven together in a three-dimensional lump that much better reflects the organ. So these three-dimensional biologies have been twice as predictive of safety as two-dimensional biology. Great progress there. Try to do safety prediction. There are some approaches that do that. This has been limited, unfortunately. We have taken the next step up of combining three-dimensional biology, these organs on chip, together with AI, and that has produced dramatic results that currently outperform all other approaches to the best of our understanding.
Thank you. So, if we think now of the applicability specifically to rare diseases, you
Yes.
mentioned earlier about the ineffective positioning of current methods and trial designs for that. Can you comment on AI's applicability there?
Sure. So rare diseases is really a passion of mine and of our group, and so we've realized that by using a combination of three technologies, the two that I mentioned, organ on chip and AI, together with a third that I'll mention here of stem cells, it is possible to not only create a replica or a model of a human tissue, but also a model of a human tissue of an individual together with the genetic ailment of that person, if this is a disease that has genetic aspects to it, or by knocking out certain genes and so creating the disease model within this tiny brain or tiny liver, etc. And so the ability now exists on many diseases to create these diseases in a dish on these tiny miniaturized human organs, especially when this is stem cell based. That's very powerful because, for example, my colleagues and I have done work on FragileX, a cause of a rare disease that's the most common hereditary cause of autism and cognitive impairment because of a single gene that's inhibited in the embryo epigenetically. By creating the brain of an individual with FragileX, you can actually mimic the disease and hence screen for drugs that hopefully remedy it. We've shown in our work a step further that by combining several such different organs, you can actually really recapitulate how a suggested drug behaves in the overall body. Hence, we call it patient on a chip. We combine, for example, liver on a chip, blood-brain barrier on a chip, and brain on a chip, and the combination of these three organs allowed us to screen drugs and to know which ones will work or not work in the human body, hence doing a better job than what was possible until now with animal testing. In FragileX specifically, there isn't even a disease model in mice. For that diseases, some rare diseases, they're typical for human and they do not exist in other animal models. And so using that in combination with AI has been very powerful. It is a long road, but very promising results because of the ability to harness the best biology that's currently known to man, which is these miniaturized human organs on a chip based on stem cells so that they reflect an individual, not just a brain of a human, but the brain of a human who suffers from a genetic disease such as FragileX or other things, and the power of AI, which allows us to run thousands of these experiments on many known drugs and then train the AI to recognize what would constitute a toxic reaction or a non-toxic reaction so that when a new drug is evaluated, it's actually the AI that is doing the heavy lifting of the prediction of safety and the toxicity, which, as you mentioned, the low-hanging fruit is actually not a good name. It's a major problem to be addressed.
It's fascinating stuff. I mean, you say it's a long journey, it's a long road ahead, but it has come on considerable leaps and bounds in a relatively short period recently. So I'm just wondering now, as my final question perhaps, what's the horizon look like for these technologies, AI and drug discovery that we've been discussing today?
So it's a great question. I actually think that we're living in super exciting times. You're absolutely correct. These technologies have come a tremendous distance in a very short time. Within a short few years, it has moved from science fiction to something which is not only possible, but is already generating market results. And I believe that the coming decade, the coming 5 years and the next 10 years will see a revolution in how drugs are developed altogether. While this is a bold statement, I do stand behind it. Just to put it in perspective, within the last decade, drug candidates in drug discovery, a large portion of these are already drug candidates that have been discovered with the power of AI. A third to a half of drug candidates that are now on their way to become drugs have utilized the power of AI. Quris.AI is leading the charge in now harnessing this power together with stem cells and organs on chip, especially patient on chip, multiple such connected organs on chip, opens the door for doing a similar revolution in personalized drugs and safer, faster drugs, and hence a deep impact on healthcare and the cost of healthcare, and a real message of hope for rare diseases.
Thank you, Isaac. It's been an absolute pleasure.
Thank you for having us. Delighted to have this talk.
And so that concludes another episode of the pharmaphorum podcast. You can find more information about this episode, including a download link and information about previous installments at pharmaphorum.com/podcasts. The pharmaphorum podcast is also available on iTunes, Spotify, Amazon Music, Podbean, and pretty much wherever you get your other podcasts. Just search for pharmaphorum and press subscribe. Of course, don't forget to visit our website itself, where you can sign up for daily and weekly news and analysis bulletins. And do follow us as well on X or Twitter and LinkedIn at pharmaphorum. That's all for now. Thank you for listening.